Grigorios, who is a postdoc at Vanderbilt University. The team at Vanderbilt has been involved in a number of studies, Genome-Wide Association Studies, and one of the issues that had come up is how do you de-identify diagnosis codes, and Grigorios was the lead author of a number of studies where they evaluated the identification risk from diagnosis codes and developed, I think, some very interesting methods for managing those risks. And his presentation today will cover the risks they've identified, as well as the techniques that they've developed. So I'll hand it over to you, Grigorios, and I'm looking forward to the presentation.
Thank you, and thanks everybody for attending this talk titled Anonymizing Electronic Medical Records for Validating Genome-Wide Association Studies. Let me first start with the structure of this talk. My talk will have two parts. The first part will be on the impact of sharing diagnosis codes on patients' privacy. I will talk about the privacy problem, whether popular protection methods can prevent this problem, and then I will present a case study with Vanderbilt's patient data. This is joint work with Josh Denny and Brad Malin, and you can find more information about this work in a paper published in JAMA this year. The second part of my talk will be on anonymizing diagnosis codes for validating genome-wide association studies. Again, I will give a brief overview of the methods and some results with Vanderbilt's patient data. This is joint work with Aris Goulalas-Divanis and Bradley Malin, and details about this work can be found in a paper published in the Proceedings of the National Academy of Sciences this year. Let me start with the setting. Actually, nowadays, researchers need access to large amounts of patient-level data, which may include demographics, clinical features, or genetic information. This data is useful for a number of studies, one of which is genome-wide association studies. Basically, the purpose of this type of studies is to discover associations between clinical and genetic information. For example, researchers may be interested in finding associations, relations between asthma, which is represented with a set of ICD codes, and some region in the DNA. For this purpose, organizations share data beyond their borders. For example, Electronic Medical Records in Genomics Consortium is a group of 5 institutions, including Vanderbilt, which promotes the use of electronic medical records in genome-wide association studies. Using large amounts of electronic medical records in this type of studies is promising because it can reduce both the cost of genome-wide association studies as well as and increase the scale of these studies. Furthermore, diagnosis codes and DNA collected for NIH-funded genome-wide association studies should be submitted to repositories such as dbGaP, which is a repository funded by NLM and NIH. So it is easy to see that when we have diagnosis codes about patients as well as genomic information, we need to do something to preserve the privacy of this data as it corresponds to patients. For this purpose, there are policies such as the HIPAA privacy rule or the NIH GWAS policy. These policies basically are contracts which data owners and data users need to sign, and basically define the uses, the legitimate uses of data. Apart from policies, there are the identification methods, which basically remove explicit identifiers from data, such as patients' names or social security numbers, as well as anonymization methods, which remove quasi-identifiers. Quasi-identifiers are attributes which look innocuous, such as demographics, but in combination, they can still re-identify individuals. Interestingly, researchers have shown that a large percent of US citizens are potentially re-identifiable based on combinations of these simple demographics. In our work, we consider re-identification of individuals, but we restrict our attention to data linkage based on standardized codes. For example, imagine that an institution wants to share a table on the right. This table contains no names or social security numbers, but just diagnosis codes and genomic information to support, for example, a study. However, by just linking the data based on diagnosis codes, an attacker can associate the identity of patients with their genomic information. This is done by just linking these two tables, the leftmost of which contains identified EMR data. So it is easy to see that this data linkage reveals sensitive information, and we hypothesize that this type of linkage is feasible because diagnosis codes are available in EMR data as well as in publicly available hospital discharge records. Linkage attacks on transactional data have been investigated in different settings. For example, Netflix published the movie rates of 500,000 subscribers who support research after perturbating them. However, Narayanan and others have shown that an adversary with some little information about a subscriber to Netflix could still re-identify the subscriber. Another study is the America Online incident, which involves the publication of search terms of about 600,000 users by America Online Research. Researchers have replaced the names of these users with pseudo IDs. However, this did not prevent New York Times journalists to use the search terms of the published data to re-identify Thelma Arnold, a real individual. So, having known these successful attacks on transactional data based on data linkage, we asked ourselves whether such an attack would be feasible. To investigate this, we used EMR data from Vanderbilt. Furthermore, we wanted to answer whether some popular privacy approaches could prevent this attack. We consider suppression, generalization, and the combination of both of these approaches. Having presented the problem, let me say a few words about the privacy-preserving techniques we consider. First, remember that anonymization techniques deal with quasi-identifier attributes such as demographics. For example, consider that an institution wants to release the table on the leftmost top corner of the slide. An attacker who knows that Greg is a 22-age male can use this knowledge to find out Greg's DNA if the data is released intact. This is because there is only one individual in the released data with age 22 and sex male. Suppression is a technique that proposes removing quasi-identifier values such as age values or sex values from the data to make this type of linkage less feasible. For example, if we remove all the age information as shown in this table on the right topmost corner, we will see that an attacker cannot find Greg's DNA because he is not certain about the actual record of Greg based on the knowledge he has. Similarly, generalization proposes replacing quasi-identifier values with more general values. Simply put, instead of removing age information, we will replace the age of patients with a range of values, such as 20 to 22 for Greg. This again makes data linkage less feasible. These popular methods were originally designed for relational data such as demographics, data that is modeled as relational tables. In this data, an assumption that was made was that each record corresponds to a single patient. However, our setting is a little bit different. First, we consider transactional data, data in which a patient is associated with a set of diagnosis codes. Furthermore, each patient, this set of diagnosis codes is very large. Instead of having 2 or 3 or 5 demographic attributes, we have thousands of different diagnosis codes, and also a patient is associated to a large number of records. So, to investigate whether the popular privacy-preserving approaches would work in our setting, we consider a population of more than 1 million de-identified patient records from Vanderbilt's EMR, and also a sample. This sample of data contains about 2.7 thousand de-identified patient records and is derived from the population we used. I would like to note that this is not just a random sample, because these records are involved in an NIH-funded GWAS study on native electrical conduction within the ventricles of heart, and this data will be deposited to dbGaP. Furthermore, the patient's profiles are of interest to many other studies in Vanderbilt. So we had to evaluate the privacy risk on this sample for practical reasons as well. So, having presented the data, let me briefly describe how suppression and generalization works in our setting. In our setting, suppression involves the removal of diagnosis codes that appear infrequently in the sample we want to release. For example, before releasing the sample on the right, we remove some of the ICD-9 codes, such as the code 401.1 in the second record. This makes data linkage less feasible because the remaining codes will be associated to more individuals. For example, if we suppress the ICD code from the second record, the code 401.0 matches to Mary and Anne, 2 individuals instead of 1 before suppression. For generalization, we simply rolled up ICD codes to their 3-digit representation. For example, instead of releasing the code 401.1 for benign essential hypertension, we release the code 401, which just means essential hypertension. The intuition is the same. Again, the remaining ICD codes may match to more people in the identified EMR data. This is the data that an attacker may have access to. So, for example, again, the second record matches to 2 records, that of Mary and Anne in the identified EMR data. As a third technique, we combine generalization with suppression by applying generalization the result of suppression. The intuition is again the same, but at this point I would like to note that all of these methods make data linkage more difficult, but may harm data utility because not all of the information contained in the original data is released. So we have to measure both privacy and the utility of the data. For privacy, we consider distinguishability, which is a simple measure that counts the number of times a certain set of diagnosis codes in the sample appears in the population. This distinguishability measure is basically equal to the inverse of the probability of associating a patient to his or her DNA sequence. For example, when we release the data on the table on the right, distinguishability of the codes for the first contained in the first record is 2, because the code 333.4 appears twice in the population. This means that basically an attacker has 50% chance to associate gene with or Jack with their actual DNA record. For utility, we consider the percent of retained diagnosis information, and I will also describe some of the diagnosis codes that were retained after we applied the privacy techniques. The first main result is the privacy risk for original data, and as you can see in this figure, we have found out that more than 96% of patients are uniquely identifiable when the data is released as is. So this result is quite alarming, and we then applied suppression to see how and if things would get better. We applied suppression using different thresholds. As you can see, the number of people, the percent of people that are uniquely identifiable gets lower, but does not reach zero, so data is still not safe unless we apply suppression with a threshold of 25%. This simply means that we need to remove all the ICD codes that appear in at most 25% of the records we release. This is enough to prevent unique re-identification. For generalization, things were similar. Generalization, when we applied generalization, still 95% of patient records were uniquely identifiable. So generalization did not do a better job than suppression. For example, the contribution of generalization to reducing the privacy risk was less than 10% in all cases. So basically the main message is that suppression was effective only when applied with a large suppression threshold, and generalization was not effective. This is attributed to the type of data we consider. The type of data we consider is sparse, which means that a patient is associated, is diagnosed with only a very small fraction of the entire set of ICD codes in the released data. This is what makes data difficult to protect with the techniques we applied. In terms of utility, our results were not very encouraging. For example, this table shows the percent of retained diagnosis information when we applied suppression with different levels. As you can see, to achieve privacy by using suppression with a threshold of 25%, only 0.12% of 5-level ICD-9 codes were retained in the original data. So the vast majority of codes, even 5-digit codes or 3-digit codes, were lost from the data. To see what this means in practice, this is the data that would be released if we applied suppression to protect the data. This data just contains 5 different ICD codes out of the thousands that our sample originally contained. And additionally, I would like to say that these codes are not useful for Genome-Wide Association Studies because they correspond to frequent symptoms. So basically, the message is that re-identifying patients using diagnosis codes is something feasible according to our study, and that some popular protection strategies are not very effective at preserving privacy and utility. Preserving privacy alone by suppression is something feasible, but the result is practically useless for Genome-Wide Association Studies. So there is a need to come up with better methods that would preserve both privacy and utility. However, I would like to emphasize that there are protection policies in place. So access to diagnosis codes and genomic information is not free for all. Basically, there are policies in Vanderbilt as well as DBGAP, the repository where this data should be deposited into, that prevent individual re-identification. However, these policies offer no formal privacy guarantees. So, this completes the first part of my talk, and having presented the problem, I'd like to say a few words to briefly describe a method that can anonymize diagnosis codes for validating Genome-Wide Association Studies. This is the first attempt to solve the problem we have presented. So, I would like to state the objective of this study, which was to prevent data linkage between diagnosis codes, the data linkage based on diagnosis codes to preserve privacy, and also to retain the associations between diagnosis codes and DNA sequences that are needed to perform genome-wide association studies. For example, our method should not allow the data on the left to be combined with the data on the right based on ICD-9 codes, and this would achieve privacy. Additionally, it should preserve the associations between the diagnosis codes and some regions in the DNA to achieve utility, to make the data useful for studies, for example, that study how many people are who have some ICD codes are associated with a certain region in the DNA. So our approach has three main steps. First, we need to find out sets of diagnosis codes that are potentially identifiable. All of these sets of codes together are called privacy policy. For example, the codes that are contained in the third record of the table, 493.00, 493.01, and 493.02 are all related to asthma, and we consider them as potentially re-identifiable. They are included in the privacy policy. Having done this, we want to find a set of associations between diagnosis codes and DNA sequences that need to be preserved to achieve utility for GWAS. All of these sets of associations are called utility policy. Again, these are sets of codes that correspond to some diseases. For example, the set of the three ICD codes in the first record of the table at the bottom of the slide correspond to asthma and are part of our utility policy. Note that a patient with any combination of these codes is diagnosed with asthma. Then, the third step of our approach involves modifying diagnosis codes in the privacy policy so that they cannot link to a small number of patients. This will achieve privacy. Additionally, we want to preserve the selected associations in the utility policy to achieve utility. For example, given the data on the left, we want to modify the diagnosis code by using a by generalizing them. This replaces all of these codes with an item denoted with brackets. This item contains all of the ICD codes related to asthma, which are the colored ones here, and it means that a patient can have any combination of these codes. It can have, for example, 493.00 as well as 493.01. Note that this patient is diagnosed with asthma when it has this this special item. So, the overall architecture of our approach is as follows. We get some de-identified records which were derived by combining records from EMR with DNA records that most of the times come from research, and apply our method to extract a privacy policy to find out what needs to be protected. Then we derive some utility policy which basically says what needs to be retained to achieve utility, and then using the data and the policies, we anonymize the data to get some protected and useful data set that can be released. First, let me briefly explain the privacy policy extraction or PPE algorithm. This algorithm gets some data and some diagnosis codes that are deemed as potentially identifiable. I will explain more on this later. And what it does is to find a set of codes that are potentially identifiable but correspond to less than K patients. This set of codes is basically what needs to be protected. This algorithm extracts this set of codes and also does some post-processing to reduce the sets of codes that need protection. One important point is how we select what type of diagnosis codes are deemed as potentially re-identifiable. There are many ways to select these codes, but we have come up with two concrete examples. The first case is called single visit case. In this case, the set of ICD codes for each patient's visit is assumed to be uniquely re-identifiable. So, when a patient goes to an institution to be diagnosed with some diseases, in a visit, all of these ICD codes need to be protected. We restrict ourselves to these codes because attackers using discharge summaries cannot use sets of ICD codes that span 2 or more visits. This has to do with the form of data in discharge summaries. However, some people would like to be more conservative and prevent attackers from knowing all of the ICD codes of a patient when data is released. For this purpose, we consider the all visits case, which assumes that all ICD codes of a patient are treated as potentially identifiable. However, our algorithm is independent of the diagnosis code selection. So, if for some studies people want to just protect some diseases, which are just the diseases that they want to release deposit, for example, to dbGaP, this is also feasible. So, a small example of applying our privacy policy extraction algorithm is this. We consider the single visit case, so we just consider simple records in the data on the left and diagnosis codes related to asthma. By doing so, the privacy policy extraction algorithm finds the colored sets of codes and then does some post-processing to retain just one set of codes, which is what needs to be protected. Then, for the second step of our approach, we wanted to specify a utility policy, which basically says what is useful for our purpose. For this study, we selected a number of diseases that are relevant to Genome-Wide Association Studies, which is the application at hand. We got these diseases from a publication from Terry Manolio, and there are 19 diseases, and each of these diseases is mapped to a set of ICD codes. For example, all these 3 ICD codes are mapped to asthma. Then, having constructed the privacy policy as well as the utility policy, we use them in our utility guide anonymization of clinical profiles, or simply UGA-CLIP algorithm. This algorithm gets privacy and utility policies as well as the dataset and outputs an anonymization of the dataset such that each record is associated to at least K patients. So it guarantees protection. How it works? Briefly put, it considers sets of codes in the privacy policy in a recursive in an iterative function. It gets a set of codes that needs to be protected, and then it applies generalization to the ICD codes. The way the generalization is applied is according to the utility policy. So basically, we generalize together things that would still be useful when generalized. Furthermore, we generalize semantically close diagnosis codes. So we put, for example, one ICD code with asthma, one ICD code that denotes asthma, together with another ICD code that denotes asthma, instead of with an ICD code that denotes heart attack, for example. By doing so, there are 2 cases. One case is to achieve privacy. So when generalization alone achieves privacy, we are done. And then we can select another set of ICD codes to protect them. However, there can be the case that generalization alone does not achieve privacy. This, for example, is the case when the utility constraint, the utility policy is too restrictive. For example, when a person wants to say, okay, give me data that is same as the original data. This is what I consider useful. This is obviously something difficult to do in practice. So in this case, we chose to preserve privacy at all costs, and we suppress code. So by suppressing code, we will always achieve privacy, but we may violate some parts of the utility policy. So the data may not be useful for a small number of the diseases included in the utility policy. As an example, consider somebody that wants to release the sample on the left. Assume that we have found a privacy policy and a utility policy, and we applied our Yuga-Clip algorithm with k equals 5. The result is denoted at the bottom of the slide. As you can see, each set of codes in the privacy policy appears at least 5 times in the resultant dataset. For example, the codes for asthma 493.00, 493.01, and 493.02 appear 6 times. This achieves privacy because it makes data linkage less feasible. Furthermore, the associations between sets of codes in the utility policy and DNA sequences are preserved. This is because the number of patients with asthma is the same as in the original data. You can see that whenever a person has this item in brackets, i.e. a combination of these diagnosis codes, would always be associated with asthma in the original data. So this is what our method does to achieve utility. It preserves the associations between these codes and DNA sequences. So let me briefly describe some of the experiments we have performed to test the effectiveness of our method. We have used the same data I was describing before that contains 2,700 de-identified patient records, and another dataset which is a subset of it. This dataset is comprised of 1,300 records, and it simulates a scenario in which data owners select only some specific diseases to be used as controls in studies. We have applied our PPE algorithm to extract policies automatically from the data, and we manually constructed the utility policy by getting these 19 diseases and mapping each of them to ICD codes. Then we applied the 2 anonymization algorithms, Yuga-Clip, which is our algorithm, and A-Clip, which is similar to our algorithm but does not take into account utility policies. So the first result is to really see if releasing the data intact would again be something risky. As you can see in this graph, the risk is always quite high if data is released intact. It is more than 95% in the all visits case, and more than 40% in the single visit case. So we have to do something to preserve privacy. When we applied our Yuga-Clip algorithm with k equals 2, and for the single visit case, we have found that a large number of the diseases of the diseases in the utility constraint in the utility policy are preserved. This means basically that our data retains its utility for a large number of diseases related to Genome-Wide Association Studies. On the contrary, the A-Clip algorithm that does not take utility policies into account did not manage to retain any of these, to retain information related to any of these diseases. So this shows the effectiveness of the Yuga-Clip algorithm in this scenario. Again, when we consider the all visits case, we have found that our algorithm outperforms the A-Clip algorithm. So a more thorough test includes changing the K value, for example, and the results are shown here. As you can see, the Yuga-Clip outperforms the A-Clip in all cases. For example, when K equals 2, Yuga-Clip retained information related to almost 80% of the diseases, while YouGA only about 35. One thing to note is that anonymizations in the single visit case offer better utility. This is because we assume less powerful attackers. Remember that in the single visit case, an attacker knew only the ICD codes that correspond to a single visit. Furthermore, when we use the small k value, we again achieve better utility. This is because we assume attackers that are not very powerful. They cannot link data that is anonymized with k equals 2. For example, an attacker that cannot link data with k equals 2 is obviously less powerful than an attacker who can link data even when data is anonymized with k equals 25. So another aspect of measuring utility has to do with how our method can support clinical case count studies. Before showing the results, let me say that anonymized data may support queries less effectively than original data. This is because we modify the codes. We modify, for example, a set of codes related to asthma, and we release these sets of codes instead of the original codes. So, for example, one person that wants to learn the number of patients that have a set of codes that appear in many records, which is something useful for epidemiology and data mining applications, may not get high accuracy when performing this study on anonymized data. For this purpose, we evaluated the number of records that are retrieved incorrectly as the result of a query when this query is applied on anonymized data. So, as you can see in these two graphs, we have found that our algorithm YouGA is comparable to the ACLIP algorithm. This basically means that data is still useful for studies beyond those included in the utility policy, for studies beyond Genome-Wide Association Studies. So, the main result is that by tuning our algorithm to help GWAS studies, we do not lose utility for different studies, such as for clinical case count studies. So basically, our anonymization approach is the first to offer formal privacy guarantees against data linkage based on diagnosis codes. And it also has some key characteristics. First, it automatically extracts privacy policies, which is something practical, because these policies may be difficult to be extracted manually. And additionally, it constructs anonymized data that helps validating GWAS. Furthermore, it can work without modification with other types of standardized codes, such as CPP codes or SNOMED concepts. So, to conclude this talk, I would like to summarize what I've said by first mentioning that re-identified patients using diagnosis codes is something feasible, and second, by saying that policies such as the NIH GWAS policy are in place, but provide no protection guarantees. So, motivated by these results, we have developed an anonymization approach that provides privacy guarantees and additionally allows diagnosis codes to be shared in a way that helps GWAS. In the future, we would like to develop approximation algorithms that we think they will achieve a better result in terms of utility. For example, they would satisfy more of the diseases related to the data they will release will be helpful to more studies of the utility policy, and this is our first goal. And the second goal is to develop open source software that will implement our approach. So that was the talk, and thank you very much. Any questions are welcome.